Hey guys, so I'm going to go over this test review for you all. This is not going to cover everything and it's not all inclusive. However, this should give you a great starting point of some things that you need to go back and look over. So, the first thing that we talked about specifically was the cell cycle. And so, um, a lot of the stuff that we looked at within the cell cycle is you got your G1 phase, then there's a G1S checkpoint, an S phase, a G2 phase, and there's a checkpoint at the G2M phase, and that's the most crucial checkpoint before we start going through division of that cell. And then we're going to have our M phase or our mitosis phase, and that's going to be the division of the nucleus. And then we're going to have a checkpoint at the metaphase plate to see make sure that the chromosomes are lined up where they're supposed to be before we go into anaphase. And then we have cytokinesis, which is the splitting of the cytoplasm. And in animals, it's going to be make two different cells. While in plants, because there's a cell wall, we're going to create a cell plate, or in this case, a new cell wall between the two new cells that you guys saw in the onion root. Now, again, here are some functions of each of the phases here for you. So another thing we looked at is, okay, so what happens within the DNA? So remember in G1 phase, all we're doing is growing some organelles, growing the size of the cell. We're going to duplicate the DNA in the S phase, so we see the from the original line to the line at the G2, looks like we've almost doubled the amount of DNA. At G2 and M phase, we haven't maintained the amount of DNA because we've not split it into cells or two different cells, and so then when we do cytokinesis, we're going to divide the cells in half, or in this case, the genetic material, which is what you see here, and then we start back at G1 again. So, overview of knowing mitosis, meiosis, and binary fission. So, binary fission is mitosis. It's diploid, it's asexual reproduction. It's going to be found in the bacterial reproduction. And in mitosis, it's diploid, it's asexual, either prophase, metaphase, anaphase, telophase. Being able to know what that picture looks like, knowing what those phases look like, are going to be really good for you to be able to know. In meiosis, it's diploid to haploid cells. We're going from one diploid to four haploid. In meiosis and mitosis is one diploid to two diploid cells. In meiosis, it's sexual reproduction. We're only making this in sperm and egg. In meiosis, it's 1 and 2, and there's variation here. The other thing within mitosis I want to talk about too is there's also going to be variation among cells. And so, depending on what cell it is, will determine how fast they need to reproduce on, how fast they're due to. So, for instance, like looking at specifically stomach cells or stomach cells. Stomach cells are going to be needed more because there's going to be more wear and tear, and so therefore they're going to need to reproduce quicker, so that we can replace the cells that are no longer viable. While if we're looking at like neurons, they're going to be in G0, they're not going to reproduce at all, or so it's going to be within years, and so those are going to be kind of very important to be just for the sole purpose of knowing that cells are going to reproduce at different rates. Variation. So how do we get genetic variation? It's going to be through crossing over, which is in prophase 1, where we're swapping parts of genetic material from chromosome to chromosome. Another way is through the two parents are going to be genetically different. And so every time that you go through reproduction, you're going to have a different set of alleles or genetic material that is able to produce a different looking offspring. Cancer. Cancer is a good one here that we're going to be looking at, as you guys looked at with our activity. So what can cause cancer? What are some environmental causes like radiation, mutations, right? So those are all very, very big things that we looked at in the last couple of days. Also, what is involved in preventing cancer? So, like things like our oncogenes, our cancer inhibitors, like our PPD3 genes. Those are all important. Being able to kind of know what they do um, are going to be very good for you to know. Being able to compare and contrast meiosis and mitosis is good for you guys to know. Knowing that um, the pictures are going to be good to know. Knowing about cancer. So one of the things about cancer too that I forgot to put on here that I just remembered is what are typical for cancer. So what they're going to do is they're going to be, since they're going through rapid cell division, so rapid cell division, most cells in our body are going to be specialized because they're going to do a certain function. In cancer, because we're going through the cell cycle so quickly, they're going to be unspecialized cells. Okay, another cool thing about cancer is that also they're going to form what we call metastasis. They're going to move from one tissue up into a new tissue, which is where you get like the um, benign tumors or the I can't remember what the other one called, but benign tumor means it's not metastasized or I can't remember what the other one's called, but with metastasize, it goes from that tissue into a new tissue. So it's going to get bigger, and it's also going to take in the body's nutrients. So we're going to start seeing an increase of blood flow to this area. So it's going to take away the nutrients from other cells that are surrounding these tissues. Again, knowing pictures are going to be beneficial for you. Knowing that our chromosomes are going to line up, so like looking at specifically within, just drawing the pictures here, this will be like prophase. These are just short little pictures. This would be metaphase here. This is line from the metaphase plate, and I'm not differentiating between meiosis 1 and meiosis 2. You guys already have seen those pictures. Anaphase. Telophase, in this case, is the cleavage furrow. Oh. And then in cytokinesis, we have 2 new cells. Ta-da! And so being able to kind of know those pictures, put them in order, that type of stuff. The other thing is knowing that we kind of talked about this previously, but I hadn't referred to it in the last little bit, is number of chromosomes do not equal complex organisms. So, for instance, like we have 46 chromosomes, as you guys saw, like there are ferns that have like hundreds of chromosomes. We are more complex because we have more specialized cells comparatively to So we have an increase in specialized cells to the decrease in plants. And that's pretty much it. Knowing about sexual asexual reproduction is going to be good for you as well to know, looking at that chart. Knowing what could go wrong in prophase, metaphase, anaphase, telophase, like for instance, prophase, nuclear envelope not condensing, or spindle fibers not forming like they're supposed to. In metaphase, chromosomes not lining up in a straight line, so they would be pulled apart differently, so we'd have a different amount of DNA in each. Anaphase, again, unequal pooling could be an issue there. Telophase, not having cleavage further in the middle, or we're skipping this phase. Cytokinesis is not splitting of the cells, so therefore we'd have multiple nuclei in each of the cells. So I hope this helps you guys. If you guys have any questions, please let me know. And good luck for Wednesday and Thursday for our test.